Today I'll be going over a brief overview of my work in the Chain Lab, which involves using gene co-expression networks to investigate Huntington null phenotypic rescue behavior observed in Discoideum Discoideum. Huntington's disease is a heritable neurodegenerative disorder that's caused by mutations in the gene Huntington. Mutations in the gene cause degeneration in neurons. Unfortunately, the molecular functions of Huntington are currently not entirely understood, so there's very few treatments available that are effective for the disease. These are the three different cell lines being investigated in this analysis. On the left are the wild-type cells, which are the cells with no additional treatments or changes. In the middle, we have the cells with a non-functional Huntington gene, which as you can see, did not develop since nothing's there. And finally on the right, we have the cells with a rescue mutation. Despite having a non-functional Huntington gene like this cell line, mutations in these cells allow them to develop anyway, although they are not quite as developed as the wild-type cells we saw on the left. We're trying to investigate what molecular interactors are responsible for this rescue behavior that we're seeing here. So to investigate the potential interactors responsible for the rescue behavior we see, we're using a gene co-expression network like what you see here to visualize our data. Each of these circles represent a gene, and each line represents that the connected genes are expressed at the same time, aka co-expressed. The closer these genes appear to each other, the stronger their co-expression. This is an example of a module, a group of genes that are very similarly co-expressed in our samples. Our gene co-expression network as a whole is made up of several modules like the one shown here. To start this analysis, samples were collected from each of the three cell lines. These samples were then sequenced using RNA-seq. The data generated by the RNA-seq was then normalized and processed so that it could be analyzed using the R package WGCNA. The data was then used to construct a gene co-expression network, and finally modules of interest were identified through visualization of the network. In our analysis, we have identified four main modules of interest. The Huntington gene and the four mutant genes are found in one of each of these modules. Some modules also have interacting genes that are differentially expressed, which means that their expression changes depending on the condition they're in. They are either upregulated, which means that they have higher expression, or downregulated, which means they have lower expression. These modules of interest potentially indicate which interacting genes and pathways regulated by the mutant genes are responsible for the rescue behavior that we saw in the mutant cell line. Thank you so much for watching my video, and thank you to Lauren Cuoco, whose data was used for this analysis, and my PI, Dr. Frederic.